should have the same number of chromosomes that you started with in G1. So, because you're going directly into G1 after that point. So if you started with 5 chromosomes in G1, you went and made 10 chromosomes. That means you have 5 sister chromatids, but you have 10 chromosomes attached to each other. Then the cell splits, and so you end up with 5 and 5 in your 2 daughter cells, which is the same number as you started with in G1. You synthesize in S, so you prepare to do your synthesis in G1, you synthesize in S, and then you grow some more in G2. So the S phase of interphase is where you make your copies of your chromosomes. And then you go into mitosis, which is prophase, metaphase, anaphase, and telophase. In prophase, your nuclear envelope is going to dissolve, and you're also going to have those chromatin, that all those DNA that's loose in your nucleus, it's going to wind up into chromosomes. And then in metaphase, they're going to line along the equatorial plate. Sorry, I'm stuttering today. So metaphase, you're lining up across the equatorial plate along the middle. In anaphase, your sister chromatids are going to pull apart, and you are going to end up, there's a way that it's worded on here that's not something I normally say, sister chromatids split and become chromosomes. So in anaphase, that's when your sister chromatids will divide, that how I said that you would have 5 sister chromatids, but there's actually 10 chromosomes there. In anaphase, that's where that splitting occurs. In telophase, you end up with 2 daughter cells with identical nuclei, or it could be different nuclei depending on if a mutation occurred when you were copying your DNA, but different nuclei is not the goal of mitosis. And then cytokinesis is where the cytoplasm would divide. So in G1, let's say you have a normal cell, n equals 10, so number of chromosomes is 10. You start out with 10 chromosomes. In the S phase, you have 20 chromosomes with 10 sister chromatids. G2 is the same, and then again at the end of mitosis, you would go back to having 10 chromosomes in each cell. So what are the parts of chromosome? The centromere is the center where it is attached. One sister chromatid is attached to the other. That's a good definition according to your test. And then a chromatid is, trying to read your test to make sure I tell you exactly the way it works. A replicated chromosome will be the answer that you want to pick for a chromatid. It's a chromosome and its copy, so a replicated chromosome. Then you have it where you need to know isotonic, hypertonic, hypotonic, and their solutions effects on animal and plant cells. So on your test, you have a couple of questions that ask you how would a cell act if it were placed in different solutions. So if you put a red blood cell into an animal solution, if you put an animal red blood cell in a hypertonic solution, that means there's more stuff on the outside than there is on the inside, the water would leave that red blood cell and it would cause the red blood cell to shrink or shrivel up. If you put that red blood cell in a hypotonic solution, which there is less stuff outside than there is inside, which means more water outside than inside, the water would rush into the cell and cause the cell to swell and eventually rupture or burst. In an isotonic solution, the cell would just remain the same. It wouldn't do anything. In a plant cell, it would cause the plant to wilt, because if it were in a hypertonic solution, the cell membrane would pull away from the cell wall, but it wouldn't totally shrink all the way down. That's what causes a wilting, is if the central vacuole is decreased. So water is leaving the cell, it causes the central vacuole to decrease in size and the cell membrane to pull away from the cell wall. Plant cells, however, would never rupture because they do have their cell wall, so eventually osmotic pressure would level out when it gets as full as it could be, and water stops rushing in, and so it maintains the same size. Then you have your laws of thermodynamics. I did talk about this in your notes, so I hope you picked up on a few of them. It asks you about your first law and your second law in thermodynamics. Your first law of thermodynamics says that energy cannot be created or destroyed. It can only change form. And then in your second law of thermodynamics, it talks about how everything's always going towards a state of chaos, which is entropy. So things are always going to break apart in nature naturally, but they're never going to join together without addition of energy. So if you're not adding any energy, the system will eventually go into chaos. So we constantly have to maintain ourselves, maintain our rooms, maintain our everything in our lives. It requires energy to maintain a state of order. Otherwise, we go into entropy. So one of your questions ask you about the second law of thermodynamics, and you're looking for the answer is the useful energy in a system will decrease as energy is converted from one form to another. So as it goes, think of glucose. You've got a lot of energy stored in glucose, but then it breaks into different forms. You've got it in carbon dioxide, you've got it in water, you've got it in ATP, and some of it is lost in heat. So as energy changes from that chemical bond in glucose, you're losing some of it to heat. In an ecosystem, you're actually only gaining 10% of the energy that was available from one organism to the next as you consume that organism, whether it's an animal consuming a plant or an animal consuming an animal. Only 10% of the total energy is acquired, able to be used. The rest of it is lost as heat and in other parts of reactions. You lose some of that energy as you go. So the useful energy is decreased as it is converted from one form to another. And then so entropy is chaos. I think there is a question about that, but I'm not finding it right away as I go through it. If I find it, I'll come back to it. Okay, moving on. ATP, what are the parts and what is it to a cell? So I'm going to actually make my life a lot easier. So ATP is composed of adenine, which is your DNA base, your nitrogenous base, sugar, and 3 phosphate groups is what you're looking at. The T stands for 3. So you've got your adenine, your sugar, and your 3 phosphate groups. And what is it to a cell? It's your energy currency of a cell. It's basically like a savings bond of energy for the cell. It's an immediate source of energy if you are building stuff. Active versus passive transport. So I'm going to actually type, I've controlled F. If you don't know what that is, you can find things in a document really easily, and it makes your life a lot easier. Oops, ACTIVE, and so you can search for stuff in your document. It's not working for me for some reason. Give me just a second. There we go. So it wants examples of active transport on your test. So active transport includes pinocytosis and exocytosis, receptor-mediated endocytosis, and or phagocytosis are examples of active transport. Also, your sodium and potassium pump are active transport. Things that are passive transport include osmosis, which is a type of diffusion, facilitated diffusion. So when you're doing passive transport, you're going downstream, and when you're doing active transport, you are taking in energy to make that happen. You have a question about the activity of many enzymes being controlled by the addition of a phosphate molecule to an active site. Oh, that's not active transport. Never mind. We won't do that. The other thing you have to do with active and passive transport is a matching, and I think that's down at the bottom. Here it says match the following descriptions of cell transport to their descriptions. Match the following cell transports to their descriptions. So you've got simple diffusion, facilitated diffusion, active transport, osmosis, and then facilitated and an active transport. And gas movement, such as carbon dioxide and oxygen, occurs by simple diffusion. If you are moving a molecule across the plasma membrane by ATP, ATP hydrolysis is the word it uses on your test, but if you are donating a phosphate, using that ATP molecule, it's always active transport. Water will move across a plasma membrane down its concentration gradient from high concentration to low concentration is what that means through osmosis. Phagocytosis is a type of active transport. It's actually endocytosis, so you're bringing stuff into the cell using the cell membrane. A molecule of a substance requires a carrier or transport protein. So active transport also could use a transport protein as well as facilitated diffusion. Both of those would use carrier or transport proteins to move stuff across the membrane and assist in that movement. Sometimes you have to have ATP to do so, other times you do not. And the last one, it says molecules move without aid and move down their concentration gradient. That's simple diffusion. If you're moving without aid and you're going down your concentration gradient, you are simply just diffusing across that membrane. All right, I'm going to stop these notes here, and then I'll pick them back up with the second part later.